Hello and welcome. We're glad you've joined us for this live webinar, Why Aren't You Seeing Under the Microscope? How New Leica Stereo Microscopes Can Drive New Discoveries in Your Lab, presented by Dr. Heinrich Bürgers. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website, and sponsored by Leica Microsystems, a world leader in providing innovative microscopy, camera, and software solutions for imaging and analysis of micro, macro, and nanostructures. Before we begin, I'd like to remind everyone that this presentation is interactive. We want to hear from you. So questions and comments can be submitted via the green Q&A button at the lower left of your screen. We'll answer as many questions as we have time for at the end of this presentation. Also, you're viewing this presentation in a slide window. To enlarge that window, click on the icon in the lower right-hand corner of your slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button and let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your web page and follow the process for obtaining your credits. Without further ado, I'd now like to present our speaker today, Dr. Heinrich Bürgers. Dr. Bürgers is a product manager at the Stereo and Digital Microscopy Division of Leica Microsystems based in Heidelberg, Switzerland. He studied biology at the University of Regensburg and Munich in Germany. After receiving his PhD from the University of Heidelberg in Germany in 2009, he worked in the field of image analysis and fluorescence slide scanning before joining Leica Microsystems as a sales rep in 2011. In 2014, he took on the role of product manager in Heidelberg and is now taking care of the fluorescence stereo microscope line. Dr. Bürgers will now begin his presentation. Welcome, sir.
Hello, and thank you for joining this webinar where I want to talk a little bit about what we can see in the microscope and how this can help us to make better decisions in research. When I'm talking about decision making in research, I'm not talking about the long thinking process. I'm talking about how to make a decision which fly to use, which worm is the right one, the left one, the right one. Is my fish at the right stage? All these decisions we all make all day in our brain nearly intuitively. And these decisions are based on what we see under the microscope, under the stereo microscope mostly. And we need to make sure that we all see the same details to have the same understanding. So in our laboratory we have to teach and we have to make sure that everyone sees the same thing and makes the same decision. This has a big advantage because if we all make the same decision, if we all take about the same thing, then we improve the probability level of our experiments, and this means we need maybe less repetitions and we have maybe better outcome. And if you see more things, and I want to talk about innovation, where you can see maybe more things in a bigger context, even higher resolution at a lower magnification, then you may have an advantage over other microscope users which don't see the same level of detail you do. When we talk about the work steps in the laboratory where you're using a stereo microscope, then I want to put this into 3 or 4 portions. That's phenotyping, picking, injecting, and maybe we can separate these a little bit more because the one part is the viewing part and the other part is the manipulation part under the microscope. And with a better image, you have increased confidence and you can decide, can I continue with that sample? Is it the right thing I'm doing? Where am I setting my injection? Am I preparing the right thing? Then once you have injected maybe RNA, DNA, you want to go and see whether the fluorescence of your protein is expressed correctly, And then you need to decide, do I want to propagate that further, or do I want to move on to fluorescence imaging? And in the end, you want to make some imaging, some measuring, and you want to describe what's happening in detail and over time. Then there are a lot of other things happening in the laboratory where the microscope is most likely not involved, like sequencing, like biochemistry, and all these things will together then form an image, and hopefully the image which you publish in your publication is made with a Leica instrument. So let's start with C.elegans to see what we can do for you with our new products. And quickly as an introduction, what are your challenges? Well, the worms are moving. There are sometimes more than one fluorescent signal per animal. And what you want to study in detail is you want to stage the worm. You want to pick the one where you can see the gonads. You want to go for phenotyping, so you maybe want to see the vulva. And you can provide different solutions for you for this. Yes, you need a base for good contrast. Sometimes you also need a yellowish light, halogen-like, to get a better contrast. You want low autofluorescence for fluor screening, low autofluorescence from the microscope and from the base. You maybe want to inject something, then I will talk quickly about inverted microscopes, and in the end you most likely go to an upright confocal. Yeah, we have a new stereo microscope, the S9, and here you can see the S9i with the integrated camera. And talking about a microscope, what is important for you? Then I found out that most C.elegans users need a magnification that goes beyond 50 times, and here we can offer 50 times magnification. And you need a large depth of field, because not all worms are on the same focus plane, and not all petri dishes are filled equally high with agar, and this can improve your screening and sorting process. You also want a large zoom range, and the 9 stands for 9 to 1, so you can go from 0.61 magnification to 55 times, and this means you can have the entire plate in overview, and then you can zoom in onto the worm that you are interested in. Looking at the oculars, it's pretty important to have a good viewing angle so that you can look inside and that you have the right position. And I will talk a little bit about optics in a later slide. Looking at the working distance, I think that's pretty important for C.elegans, because, well, you have to manipulate your samples. You have these agar dishes and you want to open and close the lid. Here the space should be sufficient. Then you want to work with needles, with forceps, maybe with brushes to take out a single worm and put it somewhere. And when you're working with students, then you will find very often that the lenses are dirty, because if you go too close to the worm, because you need a lower working distance, then everything gets dirty because someone touches it. And the new S9 is delivered from the factory with a protection glass in front of the optics, so you simply unscrew it, you wash it, and you clean again. If we talk about the transmitted light base, then it should be big enough to hold your petri dishes, maybe more than one. And you want to maneuver your petri dish around without hanging it at some borders, which is very inconvenient, because if the worm of interest is at the corner, you don't want to get stuck somewhere. And if you want to work conveniently, it's very important that you have a good contrast that's easy to adjust. And easy to adjust means for me that you adjust the contrast once to what you want, and then you can stay at that contrast. This means if you zoom in and out, the contrast does not change too much. And you can always see the level of detail you expect. And this can maybe look like this. So there's only one knob on the side to adjust the contrast, and with a half turn you can go from bright field to dark field, and these are some examples of what you may see and want to see of your worm. Another thing here to consider is if you want to inject worms, then you need to prepare the worms. And you need to prepare the worms. Here you can see an older Leica stereo and a new inverted microscope on the left, you need to pick the worm and you need to transfer them somewhere on agar pads, and these agar pads should be sticky so that the worm can't crawl away, because you want to inject maybe some RNA into the gonads. So you go to an inverted microscope, and this can be a relatively easy to say manual microscope, but it should offer you some techniques for contrasting, and you want to see the worm also 3D-like on a compound microscope here on that inverted microscope, and this means you need maybe an integrated modulation contrast from Leica, because with differential interference contrast you need polarized light, and if you're working on plastic slides, the polarization doesn't help you too much, and then the DSC image is bad. You also need a micromanipulator and an injector and an anti-vibration table. Yeah, and then if your worm is expressing the fusion protein, most likely with GFP, you want to screen it, and you need to go to a relatively high magnification, because you want to see is the expression specific or unspecific, and this gives you the possibility to decide, should I go on with that sample or not. And maybe your worm is expressing more than one fluorescence protein, then you need to have the possibility to make an easy filter change. And making an easy filter change for me or for Leica means you maybe don't need to take your hands off the sample. You can maybe use a foot switch and use a motorized filter to switch from one filter to another. If you want to zoom or to focus, you maybe want a manual instrument, because you feel more convenient with these manual controls. And if you prefer to switch your filters by hand, well, that also works with a motorized filter revolver, because you just give the filter a little push. And that's why we invented this new black guy, which you can see here, this M205 FCA, and it has an extremely large zoom range of 20.5 to 1. And this means you can go from low magnification to high magnification. If you have such a big microscope, you maybe want to think a little about ergonomy, and you can see here the ergo tilting tube on top, which allows you to adjust for taller users and users which are maybe not so tall, and always sit in a very convenient position in the laboratory where you want to screen in fluorescence. If you are looking at your worms in fluorescence and you want to just pick one, then you maybe see the fluorescence of the worm here on the left side. You see the autofluorescence from the agar and the bacteria. You see where the worms have been crouching in the bacteria. And now you find the right worm. And picking that worm is maybe not a good idea without adding transmitted light. And the transmitted light base for screening C.elegans should be possible to dim so low that you can combine the fluorescence with the transmitted light in a good contrast, because you can see here this worm is not alone, and there are at least two other worms pretty close which you could pick by accident, which are not expressing a fluorescent protein, and this means you might spoil your culture. Well, now you have the right worm, and now you go to imaging. And yes, you can do imaging with a fluorescent stereo microscope. Here on the left you see the M205FA, but more likely you will go to an upright confocal microscope, because you don't want to look through the agar, you want to look from top. Talking about the next model organism, about flies, then we have some different requirements. I've seen in many laboratories we use very different microscopes. So we use Greenhoff stereos like the S9. We use M-series stereo, modular ones, the CMOs like the M60, M80. And I want to talk a little about when to use which one. Then you maybe want to prepare something from a fly, because if you're interested in the fly brain and you don't want to image the chitin, which has a lot of autofluorescence, then you need a high magnification stereo that allows you to do this. Then I want to talk about coding. Why? Because you need maybe reliable measurements. And then we have all these implications, which mean, what does the microscope need to offer to you? A large field of view, because you want to get an overview over the flies on the fly pad. You want to see enough detail to discriminate between new mutations, others, female, male. So you can decide which flies to use. You maybe want to look at bristles and measure the length, or you want to look at wing size if you're population genetics. And this means you may use different microscopes for all these. So looking here at the work steps in the laboratory, this looks pretty similar to the C.elegans. We have still the phenotyping, picking, and preparation. You always have to decide, can I continue with that sample? Is this the right fly? And then here, in addition, you need the precise preparation for imaging. Then you need fluorescence screening. Yes, you want to see is the fluorescent protein expressed, but you also maybe want to position your sample before you go to confocal in an advantageous position so that you get a good viewing angle on the confocal, something which is maybe later not so easy to correct for. And if you can do this at a higher precision, then you have a higher reproducibility, and then you have an advantage because you need less repetitions. Work is more fun. And in the end, yes, we want you to publish. So what is important for Drosophila? And here we need multiple incident lighting contrasting methods. And you need a magnification that goes up to 40 times, usually for fly pushing. And here's something that I think is interesting. We want to talk about parfocality. So if you zoom in and out with a microscope, then you want the sample to stay in focus. You don't want to readjust the focus all the time. That's pretty boring, and it's distracting, and it doesn't help you. So what you do is you have to adjust the oculars correctly to the dioptrin of your eyes. So for me, for example, I have 1 dioptrin on the left eye and 0 on the right one. So before I even sit down, I check the oculars, set them up to my dioptrin, and then I can start working. And then I focus at low magnification, I zoom in, and everything's sharp. And it's relatively easy to adjust the oculars to your needs. So you set the diopter to 0, you focus at a low magnification, you zoom in, you adjust the diopter, and then you leave the diopter like that. And remember that position, and then it will be much more convenient for you to work. For flies, it's important to have enough working distance because sometimes you want to look at the vials where the flies are inside because you want to look at the larva of the flies. You need a large depth of field. Yes, we will talk about more in detail about the depth of field of the microscope of the new S-series, which we can provide. And that's because flies are much bigger than worms. They are much more three-dimensional, and you want to see from top to bottom, and you want to see without having to focus through the fly. On the base, you need enough space for the fly pad, and you definitely don't want cables on the desk because you don't want that something gets entangled in the cables and is pulled down from the table. We offer a lot of different bases for different applications and different flavors. And here you can see our new S9 series. On the left, the S9E. In the middle, the S9D for documentation. On the right side, the S9I with the integrated 10 megapixel camera. And you can see here some users may want a very small base to save space. Some want a medium-sized base so that they can put their hands on. And some want a base with integrated illumination. Also, talking about illumination, we can see that you have different options. We have the ring lights, we have the goosenecks, and we have some more. And that's what it looks like maybe in a typical fly lab, and that's what I'm talking about, cables and goosenecks. So nothing should be in your way because there's everything full with vials, with ethanol bottles with the dead flies inside, rubber bands, etc. You have your CO2 hoses, and if we would have now additional cables for all the illumination and light guide, this would not be a good idea, I think. And this is why we started at Leica to make our own lamps, our own microscope lamps. And I want to give you a very brief overview over what you can expect. So we have, for example, the ring light, where the light comes from the top, and you can switch on and off different segments of ring lights to improve the contrast. You can use diffusers because ring lights often on the shiny surfaces of flies make these patterns. Some users like spotlights, goosenecks as they are called, better because you can make a very nice contrast with them. You can position them freely. The little disadvantage with goosenecks is when you think the illumination is correct, then the goosenecks are usually there where you want to put your hand. So we have maybe a better solution which we can offer you. Backlight illumination, well, there are lots of different bases which can provide light that comes from below. Uniform light, if you want to check the color of your slides. Directed light, where you can adjust the contrast. And I have here in the talk our new 2 LED bases. Diffuse illumination, well, if you ever played with the reflexes you have from the ring light, and the diffuser was not enough, then we have a very high diffuse illumination, highly diffuse illumination that allows you to take shadow-free images. That's more for imaging and less good for preparation because it impairs a little bit the working distance. But for imaging and for documentation, that's maybe a very cool solution to think about. Then we have the multi-contrast illumination. That's one of my favorite illuminations because it provides the same illumination contrast like a gooseneck, but it's not always in my hands because light comes from a relatively flat angle from slightly behind the microscope, and I can adjust the segments so they're higher and lower and a little bit closer together maybe, and then I get a lot more light exactly where I need it. And this is also bound to the focus of the stereo, which means if I change the focus position of my microscope, the lamp also moves up and down together with the microscope, which I think is pretty nice because then the light is always there where I want. And then last but not least, we have the near vertical illumination. This is where the light comes pretty close to the optical beam path, so nearly through the objective. We also have a solution where the light goes through the objective. So for small animal surgery, for example, that's cool illumination because it can deliver you the same lighting conditions as, for example, a dentist or a ear, nose, throat physician would use for his patients, and that's pretty cool illumination which you can use. That's what they could look in reality. So you have the spotlights, the diffuse illumination, the multi-contrast ring light with all the adapters, polarizer and diffuser, and here's a very easy transmitted light illumination, the BLI. Here's another solution where everything is integrated. And we started with this some years ago, and we found out that this is a pretty good solution, I think, for Drosophila labs, because here you have a 4-point ring light, and you have this arc in the back where you can switch on all LEDs or just the lower one, and this allows you to play with different scenes. So light from above, light from the back at a high angle, light from the back at a very low angle, so you can see even smallest details because they are nicely contrasted. Bristles sometimes are much better to see if they make a little shadow. And everything focuses together with your microscope, and the LEDs are arranged in a way that they don't make too much reflection. So they are not too close, not too far away, and they are always illuminating the field of view where you're working. And we take a lot of effort to get the colors of the LEDs right so that you see nearly realistic colors as good as we can achieve with LEDs, and that you always see the reality and make the right decision when it comes to, for example, finding out different colors in population genetics of flies. So what's important for the flies is you need to move space to move your sample around. You need space for your fly pad. No borders where the fly pad hangs. Good LEDs for good color. The light should not reflect too much. We cannot completely eliminate this, but we can do our best. Incident light base that produces contrast. That's interesting. That's what I just showed you, the LED 2000 stand. And here's a question about, do we need transmitted light for Drosophila? And I think some of you are working with Drosophila larvae, and I've already shown you the new TL3000 ergo base before for C.elegans, and we can also use this for Drosophila. And this is where we want to play the movie, in which you can see, hopefully, a Drosophila larva, which I put in a droplet of water, crawling around, and you can see nicely internal structures like nerves, hopefully muscles, etc. And I played a little bit around with the contrast to show you what you can achieve with a base. Can we play the movie, please? Okay, I hope everyone was able to see that we can see nicely internal structures of that larva, and maybe you're interested and want to have a demo of that base, and we can come back to you later. When we're talking about flies and different microscopes and illumination techniques, we also have different microscopes which I see in many different laboratories, and there are in principle two different kinds of stereo microscopes. Both were invented in the US, the one more than 100 years ago, the other not too long ago. And here we have two completely different principles. So on the right side we have the so-called Greenhoff type, on the left side we have the so-called common main objective. And I want to explain you a little bit more what the difference is before I explain you when to use what or when to consider what. So on the right side you see the Greenhoff design, and here you see you have one microscope for every eye. That's a pretty cool idea. So you simply take two small microscopes, put them together, and then every eye has its own microscope. You put them together in a certain angle, and at Leica we look a lot at the human physiology, and we want to look at viewing physiology so that you get a really good 3D image. And the angle between these two microscopes is in the Greenhoff S9 around 10 degrees, which corresponds most likely to the viewing angle when you're reading a book. So if you look into the microscope, it's pretty easy to synchronize the two images in the brain and to get a very good, very quick 3D impression. On the left side we see this common main objective design, and you see there's a big lens, the common main objective. And above we have the zoom. Again, not two microscopes, two zoom parts. Above this we have the tube where the image recognition happens, and then we have the eyepieces. And the common main objective focuses the two beam paths onto the sample. You can imagine if you take such a big lens and you go through the outer part of the lens, then this is maybe not so cheap to make, but I think this is the possibility where you can get the higher resolution. I will show you. So the first advantage of the Greenhoff design on the right side is the working distance. You have two microscopes, you have a lot of space. You don't need the big common main objective in the front. On the left side you have much less working distance, but if you look at more detail at the CMO, then you quickly realize that, okay, now I have the possibility to exchange objectives. So on a CMO, you can have an objective revolver, because above of the common main objective, you have an infinity part. You can also add illumination there, or focus with the objective only. Then above we have the zoom body, and above the zoom body we have another infinity space, and here you can add a lot more accessories. So we think about fluorescence. Here you would place your fluorescence axis. We think about ergonomy. So here you would exchange the tube so that you get an ergo tube. We would think about coaxial illumination for material science. Yeah, here you can play a lot around. And here you see the advantage of the CMO design is the much higher versatility, and in some areas also regulatory, because if you're suffering from neck strain because you always have to look in the same angle into your green-off microscope, then this can easily be resolved by taking a CMO with an ergo tube, which you can adjust to your needs if you're working for hours. There are other advantages and disadvantages of the green-off and the CMO. Here on the left side you see the green-off, and you have two microscopes, and every microscope has its own focal plane. And if the two beam paths are tilted in 10 degrees, then also the focus planes are tilted. And on the right side, if you go through a common main objective, then everything is in the same focus plane. We can enlarge here the green-off stereo microscope, and you see in the middle the yellow where the two beam paths from the left and the right eye come together. Everything's sharp, but if you look at the left side, then the left eye and the right eye have different focus planes. And this can lead to effects like this. So the fly in the center is always sharp, and the fly at the border is unsharp, at least for one eye. Whereas with the CMO, you have a better overview. So why do many Drosophila and C.elegans users use green-off stereo microscopes? And there is a different way how they treat their samples. With C.elegans, you go to high magnification, so you put the worm in the middle, in the center of the field of view, and then you zoom in, so you don't have the problem. And the same is for flies. So there are laboratories where flies are pushed with the brush, and there are laboratories where the complete fly pad is moved around. So if you always move the fly pad so that the fly is in the center, you don't have a problem. But if you want a large overview and do quick virginal and male separation at low magnification, then a CMO is maybe the better solution. Well, we know that many users still like the green-off better because of the physiological viewing angle, and this is why we have invented FusionOptics. What is FusionOptics? Okay, so if we look now at a CMO microscope in that case, and we have unscrewed the lens, then you can see two identical pupils or entry lenses. And here we already see a problem. You can't increase the resolution any further because the glass is already touching in the middle. But we can make one big and one small entry pupil for the zoom. So on the right side, for the right eye, you have a high resolution, and on the left side, you have a lower resolution but more depth of field. And our brain will put it easily together. I mean, we have two eyes, and these two eyes never see the same thing. That's why they are in separate places in the head. And they always put together what they see. And this can help you to improve the depth of field and the resolution in one go. And this is what it looks now like on the new S-series stereo. On the right side, you have a large entry pupil for a very high resolution. On the left side, you have a smaller, in this case objective, for the left eye for the larger depth of field. And this means everything looks better in focus, it's easier to distinguish, and then you still, for fine adjustment, put your sample in the middle. Good. Next you want to go to fluorescence, and well, let's say your transaction was successful and your Drosophila brains, for example, are expressing some fluorescent proteins. Then you need to prepare them, and you need to check them before you go to confocal. You also need to screen the flies, and this is where we can help you with fluorescent stereo microscopes. And that's why we have invented the triple beam. So we not only make two different beam paths for two eyes, we not only make two different beam paths of different size for two eyes, we simply add a third beam path for the fluorescence. And what sounds funny in the beginning will hopefully easily be explained when I tell you that fluorescence excitation light does also excite, for example, lens glue. Then there's glass. There are lots of different glass in a zoom body, especially of a high-end stereo microscope. You expect 22 lenses or more in the zoom body. And these different glass, they can exhibit autofluorescence. And there can also be reflexes from the coating. And if you go through one of the optical beam paths for fluorescence excitation, this all will deliver background. And background is bad for signal to noise. And what you want to see is maybe even single proteins. And this means you need a very sensitive instrument which produces no autofluorescence. So we have added a third beam path to the stereo microscope dedicated for fluorescence excitation light. And that's in the front of the microscope. And it zooms together with the two optical beam paths. So the light is always exciting the entire field of view you're using. And if you're zooming out, it gets bigger. And if you zoom in, it gets smaller and brighter. Another very interesting thing is that you don't need a dichroic anymore, because the excitation light goes through the excitation filter and the emission light goes through the two emission filters for both eyes, and you save a lot of light with that design. If you're looking at it from the side, then you see that in the front, here on the right side, you have the excitation, and in the middle, you have the two eyes, which go to the left and the right part of the CMO lens. And the light goes through the excitation filter, comes most likely from a metal halide lamp, goes through the optics, it goes to the sample, excites the fluorescence, and all the reflexes which you have from surfaces, from plastic dishes, from glass, from liquids, these reflexes all go away from the user, back to the column. And this means only the fluorescence light reaches the eye of the user or the camera, and all the reflexes, stray light and excitation, autofluorescence will never be seen by the user. Now let's talk a little bit about zebrafish. In the zebrafish, we have again more or less the same workflow or work steps: phenotyping, picking, sometimes injecting. And we always have to decide, is my fish at the right stage? Is the culture healthy? If you're working, for example, in immunology, you want to know where do I set my lesion, my injection to stimulate immune response, for example. And then after you have decided where to do what, which fish to use, that you're maybe at the right spot, that you made the right injection, then you go to fluorescence screening. And after you have selected the right fish, you go to imaging. Here again, you want to decide what you see, that what you see is the right thing, and this means you need a pretty good transmitted light base for this. You need a large illuminated field of view because you want to zoom out, see several fish. You want a large glass plate on top of the stage because you will whirl your petri dish around so that the eggs go together in the center, and you should not hang somewhere. And then you want to start at a low magnification. You don't want to move your petri dish too much anymore because fish are swimming, and if everything's shaking, that's not good for you. So then you want to adjust your contrast, and then you want to adjust the intensity. And if you want to combine with fluorescence screening, then you want a quick possibility to switch on and off the transmitted light. And that's why we've made this new base, because here with one push of the button, the light is on or off, and it remembers the last state, even if you switch the base completely off overnight. And you can even use a small foot switch that allows you to do this with your feet, so you have hands-free operation. In the back, we have made an extra hole for space for cables. That's we found out if you put your transmitted light base into a steroid cabinet, or if you have very short desks, then this is very often a problem that all the cables come out from the back. And here we have an extra short back transmitted light base, which allows you even to be installed on small benches or in steroid cabinets. Talking about contrast of a base. Why does Leica make such a big new transmitted light base? Well, we have found out that it is important to have the same good contrast from low magnification to high magnification. And if you look at a base, then it's not sufficient to simply put a bulb under the viewing area what you want to illuminate. And this is the same on the normal microscope. The bulb is deep inside the foot of the microscope, and then there's optics, there's a condenser, and the condenser shapes and forms the light and shines at the same numerical aperture at the sample as the numerical aperture of the objective. Otherwise you won't reach full resolution. If the aperture is too big, then you get stray light, and if the aperture is too little, then you get a very strong contrast. Another thing is, if you want to do contrasting, then you think in normal microscopes about phase rings, for example, and these phase rings are usually in the back focal plane. Well, the back focal plane with stereo microscopes is very difficult, because if you zoom in and out, then the back focal plane also changes. So here we have made a lot of effort to put the contrasting element far away from the viewing plane, because as you can see on the next slide, if the contrasting element is too close to the sample, then you can see a low magnification, this black stripe as you see here in the right image. And this usually happens just when the contrast starts to get good. And this is what you see on the left side with the new base. The contrasting element is so far away from the back focal plane, so far away from the optical focal plane and in the back focal plane, so that you get a very good and even contrast. And this is what you want, because you want us to see all fish in the same contrast. Otherwise you can't make a good decision. You have to move your fish around or whatever. Then the next slide I want to talk about contrast. This is a slide which I would never put on our homepage because it needs explanation. You can see on the right side, well, most users I ask would say the right image looks better than the left image. But if you look in more detail, then you see on the right image, the black is really pitch black, and then there are white spots. And black and black, there's no difference, and what we need to see is small differences. This is where the music plays. There's white and white, and there's no difference between two white cells, so I can't look deep enough inside. I maybe cannot reveal the smallest structures here. This means you have a very strong contrast. And this strong contrast comes because the aperture of this base on the right side is very low. On the left side I have a base with a much bigger aperture, and here you can see all these nice details inside and the smaller structures. And here's my proof. These are zebrafish embedded in glycerol, and on the right side in the air bubble you see you have a very small aperture, and on the left side you see you have a very large aperture. And the large aperture of the base allows you to see more structures, more detail. And now we have a little video of zebrafish eggs, and I hope you will be able to see here on the video the heart pounding, the blood flow. You can really distinguish the individual red blood cells. And can we play the video now? And I hope you enjoy it. Okay, I'm sorry that the video was so short. We have a longer version of this video on our homepage. Just Google for TL3000 Ergo, and I hope you can see it. Good. For the zebrafish imaging, you can opt for a stereo microscope, like in that case the Leica M25 FA. And This is a fully automated stereo microscope because you maybe want to image your fish over longer time periods. And this means you need a microscope where you can control nearly every condition. So you want to control remotely maybe the zoom, you want to control remotely the shutter, you want to control remotely the transmitted light. In that case, you see here the TL5000 ergo. That base is highly automated, so if you change the zoom, it also changes the aperture like an automated condenser, and you always achieve the best contrast. This base has been optimized for high-end imaging, for high resolutions, and for fully automated tasks. You maybe want to put a motor stage, or in that case a scanning stage on top, because if you have a treatment and a mock treatment, you maybe want to image both over time at individual positions. Fish are usually not staying in one place, so you maybe want to make a Z-stack from time to time. And what you need here is low excitation power and a stereo that captures a lot of light. So it's not only about the aperture of the microscope. Sometimes you need more working distance or a larger field of view. It's also about how good is the transmission of the objectives. And I think the Leica stereo microscope objectives are pretty outstanding in that quality. Sometimes you need a 2x objective, and here on that one you can see a objective revolver that can be adjusted parfocal. So if you change the magnification, it always stays in focus, and you can continue working. And we also offer a 2x objective which can compensate for up to 5 mm water column, so you can also use the correction color to improve your image quality. And we also have a very nice video of that one on our Leica homepage. On top, you want to put a high resolution camera because you want to get all the details. And here we can assure you, you can mount nearly every camera on a stereo. We offer a lot of different cameras for different tasks. And you can also take high-end CMOS cameras without shading, no issue, to put them on a stereo. So, putting it all together, the new transmitted light base for Zebrafish fits for all stereos and microscopes. I didn't talk about microscopes today, but we also have some. A transmitted light base for routine use should work from low magnification to high magnification. And this new transmitted light base, the TL3000 ergo, supports from the 0.5x to the 2x, and also the 5x with a numerical aperture of 0.5. The high NA of a base is important, but it's not the only thing that's important. You also need the shaped light. So the light needs to have a direction, like through the condenser of the microscope. Modern LED base should remain cool, especially if you're working with zebrafish. You don't want to cook them. So LED base gives you the possibility to stay relatively cool. If you set the brightness of the LED base to 100%, it will definitely produce some heat. But that's usually not necessary because the LEDs which we use are so bright that you can run easily at 75% and then you will produce no heat at all. But I didn't want to restrict the base to lower light intensities because then it would be useless for other interesting tasks like polarization, etc. A base should produce no autofluorescence, and we achieve this on our high-end bases in two different ways. On the TL5000, I told you that we have the aperture which adjusts automatically to the microscope, and here you can close the aperture during fluorescence imaging, and then you can open it again for transmitted light imaging, and then you have absolutely no background. And on our new TL3000 ergo, we have a parabolic mirror inside, and that deflects the light in a way so that there's no autofluorescence from the LED coming back. Well, at least nearly no autofluorescence. You would need a very good camera to show this. A transmitted light base should be easy to switch on and off, maybe even with a foot switch, because you want to combine it with fluorescence. You want to look, where is my fish? Then you want to check and where is my fluorescence protein expressed? Is it specific or unspecific? And you want to decide how you go on. You need a large illuminated field of view, and this is what all our bases can deliver. And you need LEDs with good color reproduction, because you want to see the beauty of life in the original and not something artificial. And you need a lot of good contrasting methods. So like the light from above, which you can adjust with your goosenecks, from the top, from the left, from behind, you can also adjust with the Leica base. So you can have the bright field, the dark field, and the Rotterdam contrast, which is oblique illumination from below, invented by Leica some years ago. When it comes together, you see there's a lot of possibilities which you can play around. And here's what I would ask you to do when you speak to a Leica sales rep. Talk about your application. There may be solutions you didn't expect. We have many microscopes for many specific tasks, and we always have a possibility to help you get better results, to help you make better decisions, and help you to be ahead of competition one step. And our new products, they were made with you, the user, in mind, with human physiology in mind, so that we see that you see more, and that we see you as happy users. And I have shown here the new transmitted light base, the TL3000 Ergo, the new fluorescent stereo M205 FCI, the black stereo microscope, and the new screening stereo microscope DS9, which is here on my last slide. Good. Thank you very much.
Thank you so much, Dr. Bürgers, for that informative presentation. A quick reminder for all of you audience members on how to submit questions. Simply type them into the Q&A box found by clicking on the green Q&A button at the lower left of your presentation window. Dr. Bürgers will answer as many questions as time permits. So let's begin with our first set of questions. Looks like we have quite a few coming in. Here's an audience question. Dr. Bürgers, could I use this microscope with fresh materials?
With fresh materials, sure. Well, fresh materials is for me a living specimen. Yes, definitely. Or if it's freshly excised material, yes. This is why we make the very big glass plate on top, so that you have space to put a big wax plate or whatever you want to do. Fresh materials, I think, also about transmitted light, maybe organs and something. Yes, and even if you spill something, our base is constructed in a way that even if salt water is running inside, you can get it out again. I mean, if you don't soak it, it will survive. But we have checked our microscopes for this, and there are spilling borders so that nothing can get into the base, into the electronics, and destroy them. And the mirrors inside can be cleaned carefully. So I would say yes. I hope this answers the question.
I think it does. And here's our next question. It's a three-part question. How are the pictures erected in the greenotype? Are there any distortions, and how are they corrected?
In the Greenhoff, yes, there is an image erection. I didn't show the prism here in my presentation, and this can be done with prisms or with mirrors. So simply some mirrors that erect the image right before the oculars. Are there any distortions? Well, that's a very interesting question. Distortions, I can think about flatness of field, and they can be constructed so that they look plan, but since you have the tilted focus planes, you will have no advantage of this. A very interesting point about distortion is color distortions, and if you go beyond 40x, then you need definitely plan apochromatic correction. Otherwise, you will see color fringes. And these color fringes will be very prominent in a green-off stereo microscope. This is why the new S9 stereo microscope is apochromatically corrected, so that you get the best image.
Wonderful, thank you. And I think you answered part of this next question. How are the different eye distances corrected for in the green-off type? Because the angle between both beams could be changed, right?
Well, if you have the image erecting prism, then you have a space where you can adjust the interpupillary distance without changing the viewing angle. Yes.
Wonderful, thank you. We have a lot of questions coming in from our audience members. Wonderful questions. Here's our next one. How different vision of both eyes is corrected for? I'm not sure exactly how different vision of both eyes is corrected for.
Okay. I think this is related to FusionOptics, and if you have two different optics for two different eyes, then you see definitively two different things on both eyes. And the larger the entry pupil, the more light you are collecting. And this means on the right side, it usually is a little bit brighter, but you won't recognize. We don't compensate for this. It's not necessary.
Great, thank you. Here's our next audience question. How do the Green-off S9 series Fusion and CMO compare for focus of the specimens at the edge of the Petri dish? Is S9 closer to CMO or Green-off?
The S9 is definitely Green-off. The S9 allows you to see more in focus with the left eye, which allows you to have a better overview and a better vision in 3D because you see also more depth. But on the right eye, you have the higher resolution, and if you want to see something really in detail, then you should position it in the center of both beam paths. With a CMO, you have no problems in synchronizing because everything is in the same focus plane on the edge of the Petri dish or in the center.
Thank you for that. And audience members, thank you so much for your interaction. We have some wonderful questions, and we have time for a couple of more. Is the light from several LED spots homogeneously scattered through the object plane?
Yeah, I mean, that's the main reason why we started to make our own lamps. If we buy lamps from suppliers, then they maybe don't illuminate the focus plane correctly. If you exchange objectives, it also meant in the past that you had to exchange the ring light. And this is why we design our own lenses in front of the LEDs. This is why we design the angles of the LEDs, and we make sure that from in a certain magnification range, and I mean different objective range, with a different working distance range, the field of view is always correctly illuminated. So you have no dark spot in the middle, no bright cone in the middle, depending on the objective you have. If you look at one of our ring lights closely, you will see that they have lenses on top of the LEDs, and these lenses have different shape so that they really illuminate the entire field of view.
Wonderful. Thank you so much. And we have time for one more question. Our last question for now, how can one view such a video stereoscopically? Is it something like virtual reality?
No, I took one of our cameras. I mounted it on the right beam path. I closed the aperture just a little bit because the right beam path is the one with the high resolution, less depth of field. So I increased the depth of field a little bit and I pressed record. That's it.
Wonderful. Thank you so much, Dr. Bürgers, for your wonderful presentation. Do you have any final comments before we end our webinar today?
I must say thank you. These were very inspiring, interesting questions, and it shows that everyone paid attention, and I obviously hit a spot here. Thank you very much. If you have more questions, I think you know how to reach us, and we will be happy to answer more questions by mail.
Thank you so much. And I'd like to thank the audience for joining us today for their interesting questions. I'd also like to thank our sponsors, Leica Microsystems, for today's webcast. This webcast can be viewed on demand through December 1st, 2017.